The Lewis antigen system is based upon two genes on chromosome 19, FUT3 or the Lewis gene and FUT2 or the secretor gene. The proteins produced by FUT2 and FUT3 modify type 1 oligosaccharide chains to create the Lewis antigens. These oligosaccharide chains are similar to the ABO blood system. The link between the Lewis blood group system and secretion of the ABO blood group antigens was possibly the first example of multiple effects of a human gene. The same secretor enzyme which converts the LEA antigen to the LEB is also responsible for the presence of soluble AB and H antigens in bodily fluids. Lewis substance is synthesized and released into plasma where it can adhere to the red blood cells. Transient changes can occur during pregnancy, as the LEA and LEB antigens may be reduced during pregnancy, meaning they may transiently produce anti-Lewis antibodies. The production and release of Lewis substance is dependent upon the LE, H, and secretor genes being active. UK genotypes and phenotypes are shown. In the first line, the person has LE and the H gene, but not the secretor gene, and is therefore LEA positive B minus, so a non-secretor. On the second line, the person has LE, secretor gene, and the H gene, so is LEA negative but B positive, which is the majority of the UK population. Even though these individuals have the LE gene, most precursor chains are converted to LEB, so the phenotype appears as LEA negative. Inheritance of only the H gene is rare, and they are LEA negative B negative. Last is the Bombay phenotype that doesn't have the Lewis gene or the secretor, so is also LEA negative B negative. Anti-Lewis antibodies are IgM in nature and are usually naturally occurring in LEA negative B negative people. Other phenotypes rarely produce the antibody. The antibody can bind complement, causing them to become hemolytic. For identification purposes, using enzyme-treated cells enhances their detection, which means the LEA and the LEB substance protrudes less from the membrane and benefit when Duffy and MNS system antigens are cleaved. Anti-Lewis antibodies are not clinically significant, as antibodies can be neutralized by Lewis antigens on donor cells. They don't cross the placenta because they are IgM, so do not cause hemolytic disease of the newborn. Also, the antigens are not well developed in cord blood, so it is also LEA negative B negative. Finally, this table shows relative immunogenicity of particular antigens. For example, the chance of developing an anti-D antibody if you are a rhesus negative individual who is transfused with rhesus positive cells is estimated to be upwards of 70%. As you can see, the chances of an immune response to other antigens is notably less, but nevertheless possible.